The vital contributions made from the scientists Iwanowski, Franklin, and Baltimore have greatly impacted our present knowledge of DNA, RNA, and viruses. Iwanowski discovered the mosaic virus and its structure. Franklin concluded the structure of DNA, and Baltimore founded reverse transcriptase and the Baltimore classification. The work produced by some of these scientists were used as foundational knowledge for other discoveries. These discoveries have greatly impacted our understanding, prevention, and treatment of viral diseases.
Dmitri Iwanowski, born October 28, 1864, in the Russian Empire, was a microbiologist. He studied at the University of St. Petersburg in 1887, where he was sent to Ukraine to study the mosaic disease in tobacco, building on the previous work of Adolf Meyer. Iwanowski was the first to detail the characteristics of the organisms that came to be known as viruses in 1892. Through an experiment, he demonstrated that the infectious agent could pass through a filter that prevents most bacteria from passing through. The sap of the leaves infected with TMD retained the infection characteristics after filtration through the Chamberlain filter candles that were made of unglazed porcelain. He concluded from that experiment that the filtrate might contain an infectious agent and help to produce the disease. These observations led to his belief that a disease agent is smaller than any already known. He failed to understand the full meaning of his experiments due to the currently accepted scientific knowledge about diseases. Later in 1903, Iwanowski described abnormal intracellular inclusion bodies in host cells of plants with viral diseases. His description of crystal-like bodies on these plants is now believed to be the virus. Today, the tobacco mosaic virus is known as rod-shaped virus with RNA surrounded by a protein coat that causes mosaic-like symptoms. These symptoms include mottled patches of green or yellow on the leaves, brittle stems, abnormally small leaves, curled leaves, and unripened fruit. This virus stunts the growth of plants, which leads to product yield. This is the reason for the deep research and experimentation into the disease that affects many different plants.
Rosalind Franklin was born on July 25, 1920, in London, to a wealthy Jewish family who valued education and public service. At age 18, she enrolled in the Women's College at Cambridge University, where she studied physics and chemistry. After Cambridge, she went to work for the British Coal Utilization Research Association, where her work on the porosity of coal became her PhD thesis and later allowed her to travel the world as a guest speaker. In 1946, Franklin moved to Paris, where she perfected her skills in X-ray crystallography, which would become her life's work. In Paris, she also worked with Jacques Mering from the years of 1947 to 1950 and published 5 landmark papers on graphing and non-graphitizing carbon between the years of 1946 and 1949. Although she loved the freedom and lifestyle of Paris, in the year of 1951, Franklin moved to King's College and began studying DNA. Around this time, Watson and Crick were working on their model of DNA, but found themselves stuck and eventually put the work on hold. Franklin was a passionate woman. Franklin worked hard and was known for her determination. She was an avid traveler, hiker, and with a great love of outdoors who enjoyed the spirit of discussion of science and politics. Friends and close colleagues considered Franklin a brilliant scientist and a kind-hearted woman. However, she could also be short-tempered and stubborn, and some fellow scientists found working with her to become a challenge. Among them was Wilkins, the man who she worked with at King's College. A misunderstanding resulted in friction between Wilkins and Franklin, and their clashing personalities served to deepen the divide. The two were to work together on finding the structure of DNA, but their conflicts led them to working in isolation. While this suited Franklin, Wilkins went looking for company at the laboratory in Cambridge, where his friend, Francis Crick, was working with James Watson on building a model of the DNA molecule. Meanwhile, Franklin had discovered the two forms of DNA, a crystallized A form and a hydrated B form. By carefully hydrating and dehydrating her sample of DNA and rigorously controlling the humidity of her lab, she was able to get pictures of the DNA as well as obtaining precise measurements of the turns and the number of pairs between them. Unknown to Franklin, Watson and Crick saw some of her unpublished data, including the beautiful photo 51, shown to Watson by Wilkins. This X-ray diffraction picture of DNA molecule was Watson's inspiration for the clearly patterned helix. Using Franklin's photograph and their own data, Watson and Crick created their famous DNA molecule. Franklin is in the shadows of science history, for while her work on DNA was crucial to this discovery of its structure, her contribution to this landmark discovery is little to be known. Franklin's contribution was not acknowledged, but after her death, Crick said that her contribution was critical. Franklin moved to Birkbeck College, where, ironically, she began working on the structure of the tobacco mosaic virus, building on the research that Watson had done before his work on DNA. It was known that the virus consisted of protein and DNA or RNA. Watson, during his hiatus from DNA modeling in 1952, worked briefly with tobacco mosaic virus and established that the protein subunits were arranged in a spiral. Franklin's challenge was to find out whether RNA was in the middle of the spiral like a candle wick or embedded in the proteins. Franklin encountered strenuous opposition when she wrote up the results of her initial work on the TMV in late 1954. She had found that TMV particles were of a uniform length and that the protein subunits were of uniform size. She sent a draft of her article to Norman W. Perry, an plant biologist based at the Rosalind Experimental Station. Perry has supplied Franklin with the TMV for her research. He also was on the Agricultural Research Council and had approved her research funding. He objected to her conclusions. She replied respectfully, but defended her conclusion, which was right. When she went ahead and published them, Perry was so annoyed that he stopped supplying her with the virus cultures, so she had to learn to grow them on her own. Due to Perry's influence, she lost the rest of her funding. In 1954, she was given the opportunity to visit America for the first time. She had scraped up enough funding by presenting a paper on occult chemistry and ventured home with more virus samples. By 1956, Franklin and her team were ready to put together a model of TMV. Donald Casper, who came to work with Franklin in the mid-1955s, had discovered that TMV had a hole down the center of the proteins. Franklin's X-ray diffraction data indicated that the RNA winds around the inside of the donut hole in a helical groove in the protein units. The team rapidly extended their studies to other plant viruses, like the turnip yellow mosaic, tomato bushy stunt, pea streak, and a potato virus, often working with material contributed by colleagues worldwide. During the next few years, she did some of the best and most important work of her life, and she traveled the world talking about coal and the virus structure. However, just as her career was peaking, it was cut tragically short when she died of ovarian cancer at age 37. Her obituary concluded that she was a perfect example of a single-minded devotion to research.
David Baltimore was born on March 7, 1938, in New York City, New York. He is an American biologist with many key contributions being in the fields of immunology, virology, cancer research, biotechnology, and recombinant DNA research. Baltimore graduated with a bachelor's degree in chemistry from Swarthmore College with the highest honors and continued on to MIT with select classes contributing towards his doctorate, then moved to Rockefeller Institute to complete his PhD in virology. David Baltimore was awarded 1/3 of the Nobel Prize in Physiology or Medicine in 1975. The motivation behind this award was, quote, for the group's discovery concerning the interactions between tumor viruses and genetic material of the cell, end quote. This discovery was one that Baltimore made in 1970 and was titled reverse transcriptase, in which the enzyme RNA-directed DNA polymerase was shown to synthesize proviral DNA from the RNA genome of RSV. Baltimore's findings led to a spur of the other discoveries of cancer-causing agents revolving around genetic exposure. Working independently, Baltimore and Timon studied how RNA viruses replicate after infecting cells. Before David began his study regarding the synthesis of DNA from RNA, he was informed that piconovarious RNA synthesis was independent of DNA synthesis. However, some results suggest the making of cytoplasmic RNA-dependent RNA synthesis system. In 1968, Baltimore was showing a specific virus reproducing itself by an RNA-dependent polymerase that copies RNA separately from DNA. He then focused on two RNA tumor viruses that led to his discovery of reverse transcriptase. Reverse transcriptase is a process in which a viral enzyme translates the RNA genome into DNA. This discovery led to the finding of viruses' ability to insert genes into cells and modify the behavior of those cells. After the newly formed viral DNA fuses into the infected cell, it causes the infected cell to generate into a cancer cell, thus discovering that cancer could, in fact, be a genetic problem. This also led to new capability of essentially copying and pasting DNA. This discovery has not only affected the cancer epidemic, but has significantly influenced national science policy on the AIDS epidemic. This has greatly impacted science today by implementing these findings in gene therapy to help treat HIV, prevent HIV infection, and treat cancer. Baltimore also developed the Baltimore classification system, which is essentially dividing up viruses in 7 classifications. Baltimore created this virus classification based on the vital role of the translational machinery and the importance of the viral mRNAs in programming protein synthesis. This led to the emphasis of the obligatory relationship between the viral genome and its mRNA. His present research mainly focuses on controlling the inflammatory and immune responses, the roles of microRNAs in the immune system, and on the use of gene therapy methods to treat HIV and cancer and engineering immunity.
Oliver Holmes said it best, quote, "Many ideas grow better when transplanted into another mind than the one where they sprang up in," end quote. It can be concluded, based on our research, that the scientists' work was a collaborative effort rather than solely individual input. Their work was built upon each other. This can be observed through the research and discovery of each scientist. Iwanowski's work led to the discovery of the tobacco mosaic virus and its structure. Franklin's research was built on information known about viruses, consisting of protein and DNA or RNA. From this, she concluded the structure of DNA. Franklin's research led to a clear understanding of viruses and how they function. David Baltimore founded reverse transcriptase and constructed the Baltimore classification, which essentially groups viruses into families based on their genome type and method of replication. The tobacco mosaic virus, also known as TMV, would be classified into group 6 because its genome is a single strand of RNA virus. TMS replicates using reverse transcriptase to convert the positive sense RNA into DNA. These discoveries have led to the advancement of research of what we know of our modern understanding of viruses and their overall function. The primary way we can observe the research of Iwanowski, Franklin, and Baltimore is through the utilization and growth of medical attention in HIV/AIDS current epidemic, along with cancer's epidemic. Because Iwanowski isolated infectious viruses within plant cells, This gave way to Franklin's ability to distinguish and identify the function of viruses, which was expanded upon through Baltimore's research and understanding of reverse transcriptase. This knowledge is vital in understanding how viruses, like HIV and AIDS, progress through the system and infect the body's entire immunology. Through this knowledge of viruses, we know how to recognize, fight, and prevent viruses in our world today.